Thank you so much for that gracious introduction, Peter. I can't tell you how phenomenally grateful and honored I am to be here today. This is a magnificent welcoming people with everybody I've seen, from the original developers to the current developers, clinical scientists. It's just a phenomenal treat. And so thank you for that. Today, I'll be talking about clinical applications of SPM, because I really feel that this innovative tool, SPM and CON, these innovative tools really drive scientific discoveries and can lead to paradigm shifts in clinical neuroscience and precision psychiatry. So before I get into that, I would really like to take a moment to express my deepest gratitude to Carl and your spectacular team for your monumental contributions to our field in general. Your visionary integration of really rigorous statistical methods with deep theoretical insight has fundamentally transformed how we analyze and understand brain function and structure. And SPM not only set the standard for neuroimaging analysis, but also empowered generations of research to explore the brain with unprecedented precision, ease, and sophistication. So thank you all for all of your work. So over a decade ago, Bernstein and colleagues showed in JAMA Pediatrics that there was already 100% increase in ER visits for children with suicidal attempts or ideation. Then, of course, not surprisingly, the pandemic spiked anxiety and depression in adults, but also in children. And this became such an issue that the New York Times had coverage, publishing many different articles, really highlighting the mental health crisis pandemic. And the US Surgeon General and leading pediatric health organizations all got together and declared national state of emergency of mental health. And it's clear that we need urgent need for innovative precision treatments that target core mechanisms underlying mental illness. So when I step back and I think about what are the critical barriers that are really preventing us from moving the needle in mental treatments, four things come to mind. One is we're still focused primarily on remediation instead of prevention. Oftentimes we're giving these treatments a trial and error approach despite the great heterogeneity in treatment response. Many of these treatments are ineffective. Both pharmacological and behavioral treatments are only effective for 30 to 50% of the patients. Sometimes they have side effects, and of course, there's high attrition. A lot of that is because we just don't understand, to a large degree, the pathophysiology of mental illnesses. Some of the drugs, the majority of drugs for psychiatry, have been accidentally discovered. So we really need to understand the mechanisms of action of these treatments and how they can work. So wouldn't it be nice if we had a biological way to look at biomarkers, say, to identify early that ways in which we can predict trajectories of mental health problems? Could we use biomarkers to predict who's going to respond to what treatments, so we can tailor these treatments? And can we use these biomarkers to actually innovate novel treatments that really target the core mechanisms of mental illness? And can we better understand the causal mechanisms? So naturally, we went to multimodal neuroimaging, and of course, you can look at brain structure, brain function. We've heard beautiful talks today on both. We've been primarily interested in the intrinsic functional architecture as elucidated by resting state networks. And I wanted to mention that individual differences in these resting state networks, we know, shed great light into brain health and disease, but also intra-individual variation across contexts also shed light in brain health and disease. And luckily, these resting state networks are plastic, being modulated by behavioral interventions. And so I set out to do this, to try to kind of form a network, precision network therapeutic, and that turned out to be much more difficult than I had imagined. But luckily, there was SPM. So we spent years and years, decades actually, learning SPM and developing novel complementary tools that would work in conjunction with SPM to really understand the brain network organization and brain health and disease, all with the ultimate goal of developing precision treatments for mental health. So the first one that we developed was ART, and that was really inspired by Tom Nichols and SPMD, because we really wanted to understand how we can clean up the data. And that was both fMRI task and resting state. And then I had the fortunate opportunity of meeting Alfonso Nitacasanan, who developed the CON toolbox, which allows us to look at intrinsic resting state networks and analyze those networks. And then we also developed real-time fMRI neurofeedback, which we can use CON to then analyze the network modulation due to this real-time neurofeedback and the modulation of the associated symptoms in clinical populations. Now we're really engaged at creating something called the Mind Balance Training Platform. And this is a closed loop system, basically, that's designed to identify vulnerable states and then trigger just-in-time adaptive interventions. So I remember very well, many, many years ago, in the late 1990s, I remember it was my first foray into the SPM code, and I was stunned. I spent many late nights looking at the SPM code and realized how wonderful it was and how complicated it was and how much I didn't know. So I was very eager to try to understand two confounds for fMRI task and resting state. I wanted to understand how motion really affects the fMRI time series, and then later on I wanted to understand how physiological aliasing and the resting state networks were confounding. And so we hijacked SPM Global to just simply plot out the SPM global signal. And you see there in the middle XY graph, this is the global signal. So each time point is each TR. And then below that, we could just plot out the associated movement. And then that will just display how you can see the movement changing and the global signal from a time series. And then we would allow the user to specify thresholds with which we would identify outliers in the global signal and motion, which we could then eliminate in the first level GLM. And what we found was surprising. So when we looked at analyses, both for fMRI task and for resting state, we found an actual dramatic difference in when we just regress out motion versus motion plus these artifactual time points. And I'm just giving you an example. This is actually an example I pulled out from a long time ago. It's a 10-year-old child. We wanted to see if there was a default mode network operating there. So we put a default mode seed, the medial frontal cortex, and we looked at the intrinsic correlations to that seed when we regressed out motion parameters and the derivatives, so on and so forth. And then we did the same analysis, but we just we regressed out those additional nuisance covariates. And what we found is that when we did not regress out the nuisance covariates, we did not see the default mode network. But as soon as we did, you can see that the PCC and lateral parietal cortices pop up. So this is actually, that was a monumental finding for me, because when we're looking at fMRI task and resting state in clinical populations, oftentimes people move more. We're also looking at developmental work. And so at that point, there were a lot of studies showing that even infants might not have the default network, which we think actually they do have a default network. And there were studies showing that this default network actually increases in the positive correlations over the course of development. But we had a postdoc, Chen Chai, who actually looked at that very carefully by placing seeds, this is the lower left, seeds in the default mode network, and looked at how that network was changing over the course of 8 to 25 years old. And we did that with and without regressing outliers. And what we found was that there was not actually, we found an increase in the positive correlations when we did not regress out the outliers, and we did not see that after we regressed out the outliers. However, what did remain was the systematic increase in default mode network anti-correlations, which we interpret as an increase in executive function. So that was really interesting because we're very focused on developmental trajectories. The second thing that we were interested in clinical populations, there were a lot of papers coming out that illustrated there's decreased functional connectivity or activation in many clinical populations. We were interested at that time in schizophrenia and high risk for schizophrenia. So after we used a toolbox to not only reject out outliers, we also looked at things like stimulus correlated motion, which Ed Wilmore and colleagues have shown schizophrenia patients often tend to have more stimulus correlated motion. So when you're regressing out those motion parameters, it could be disproportionately moving variance from one group versus the other. So we tried to aggressively tackle all these QA problems. And what we found in that particular case was a monotonic increase of the default mode network activation and connectivity from controls relative to patients, which were correlated with an increase in psychopathology. So the reason why I give you this example is because just a little bit of QA can actually have a dramatic effect on your results. The next big artifact that we were interested in tackling was the physiological aliasing that you get when you're looking at low frequency fluctuations, like less than 0.1 hertz, which we often do in the resting state context. And so, as you know, that there was a big debate about global signal regression. Do we regress out the global signal to reduce the positive symptoms, the positive false positives due to respiration and other physiological aliasing? But if you do that, then you're mathematically mandating the negative correlations or the anti-correlations, rendering them uninterpretable. So we are specifically interested in the anti-correlations. We adopted another method of noise reduction called the anatomical contour method. And Alfonso basically included that both the ART tool and the anatomical contour method of noise reduction in this CON toolbox. Now, he started developing this a long time ago and has been systematically developing it for many, many years. And it is completely interoperable with SPM. He uses the SPM code. He saves all the data that is generated by CON in SPM format so that you can read it in SPM as well. So it's seamlessly interoperable. And we have found this to be a very successful way in analyzing brain connectivity. He just recently showed me the most stats, the recent stats. He has over 172,000 downloads, and he teaches these courses and wrote a book about it. So I think it's really exciting to me that, this was a big career move for him, and that we can actually understand what's going on after taking out the artifacts of what's happening in the brains with intrinsic brain connectivity. So we use this toolbox a lot, and we use it to analyze what can happen at baseline, also what can happen over longitudinal trajectories, and what can happen due to interventions. So one intervention that we try to do is real-time fMRI neurofeedback. And the idea here is that we identify a region or a set of regions that may be clinically relevant, and then we train people in the scanner to modulate those regions. And this particular intervention is not invasive. It's personalized because we identify the individual networks, and it's network driven rather than just region driven. It's adaptive to the individual, and the reason why I like it most is because it promotes agency. So a lot of times when you're receiving a treatment, whether it's DBS, TMS, or what have you, the patient is acting as a receiver, whereas here the patient is actually actively engaged in modulating the networks and subsequent modulation of their symptoms. And we also think that the ability to modulate the neural dynamics on a network level with the neurofeedback may be a more effective method of neuroregulation and neuromodulation involving only a single region. So the way it works is that we have people lying in the scanner, we identify their networks, and then we convert it into some kind of visual display, which you can see here is a little ball task, which our wonderful postdoc Jeff will be explaining in more detail soon. But basically, we will track, say, the default network and the frontoparietal network, and we will move the ball based on the difference between those individualized networks. So if we want to suppress or downregulate the default network relative to the frontoparietal, the ball will move up, and it will move up with a hop size proportional to the magnitude of the difference between those networks. But if we want to, if the person is daydreaming and the default network starts to rise, then the ball moves down. So basically, the people are in the scanner moving this ball with their mind, then we can use CON to look at the resting state networks before that intervention and after, and look at how that network modulation is associated with modulation of symptoms. So the other thing that we can do is use this real-time system not only to feed back and train individuals, but we can also use it to trigger. We can trigger tasks, a stimuli, we can trigger experience sampling. So here's an example where we use SPM to identify a region of the developmental network, basically the PCC in blue and SMA in red. And then we use real-time fMRI to track the bold activity in those regions. And then we trigger the stimuli for a vigilance task based on when we think the person is physiologically in a good state, meaning the PCC is lower than the SMA. We trigger the stimulus, and then if we think the person's in a bad vigilant state, then when the DMN is high relative to the to SMA, we trigger the stimulus again. So we have good states and bad states, and then the patient responds. And what we find is that when we present the stimulus triggered by optimal brain states, we got faster reaction times. So we use this kind of model to explore other tasks. So for instance, we can use SPM again to localize the PPA. We can then use real-time fMRI to detect optimal and suboptimal brain states for PPA. Then we can trigger task stimuli. And what we find is we can actually causally improve human performance and subsequent memory by triggering the scenes when they're the PPA is in a good state versus bad state. And so I thought that got me wondering, well, if we can actually kind of causally manipulate human performance by presenting stimuli in this way, can we actually use this in a way to detect vulnerable states, say a suicidal ideation or an auditory hallucination, to then trigger just-in-time adaptive interventions? And so that's what we're setting out to do now with this mind balance training platform. And basically here we have people in the scanner, and we do a multimodal deep precision phenotyping, trying to identify a fingerprint, if you will, an fMRI fingerprint that is corresponding to a particular symptom, say rumination. And then we partner with the patient to learn how to modulate that fingerprint or that feature with, say, real-time neurofeedback. And then we capture the electrophysiological correlate of that fingerprint because we're doing simultaneous EEG fMRI, which we then couple with physiological sensors to do digital phenotyping in the real world context, so that the patient can practice these mind-based interventions in the real world. And here they will get triggered just-in-time adaptive interventions when that signature is high. Ultimately, what we're hoping is the patient can recognize their own vulnerable states and initiate personalized interventions on their own. So those are the tools, and I'll just give you one example for how we can translate this into the real world. So here we use CON to look at a number of kids, 7-year-old kids, and I think there were 100 kids. And we used the resting state networks to predict the progression of anxiety, symptoms related to anxiety, depression, and ADHD. And then we replicated that in other samples. And the idea here is that if we are able to look at baseline intrinsic connectivity to predict the progression of symptoms, can we then intervene early with behavioral interventions that may mitigate those symptom progression and perhaps even avert the onset of illness. We've also used resting state networks to predict treatment outcome in the case of social anxiety disorder. And we've used the resting state networks just to look at how interventions like real-time neurofeedback can be modulating the networks with symptoms. And we also are designing a new method called biological triggered experience sampling, where we basically use a combination of CON and the Murphy, which is the real-time neurofeedback system. And then we can use it real time to identify these resting state networks, track them over time. And then we can trigger experience sampling, ask the person what they're experiencing. And then if we do that enough times, then we can build a predictive model and elucidate the neural antecedents of fluctuating mental features, which will then allow us to then perform the real-time adaptive interventions. So all of this is to say that we're trying to understand the brain connectivity in ways in which we can help people modulate their connectivity and symptoms. When we look at the data to do causal mechanistic understanding, that will help us refine the interventions and tailor the treatments. So let's see, where am I at time? Okay, so I will just say that one of our operating hypotheses here is that there's hyperconnectivity of the default mode network, which we think is a targetable transdiagnostic mechanism underlying many mood and psychotic disorders, and that this large-scale network kind of ropes in subgenual ACC in the case of depression and auditory cortex STG in the case of schizophrenia. So when we downregulate this network, and a network level, we're not only downregulating the default network, we're also downregulating this association with subgenual ACC and STG, which then subsequently is related to mitigation of those symptoms. So we also are, the second hypothesis is another version of DM and hyperconnectivity, which is just basically a lack of anti-correlations. And so we think that reduced DM and anti-correlations is also a targetable biomarker of cognitive impairment. And we've specifically been looking at the magnitude of the MPFC and DLPFC anticorrelations. They seem to rise over development selectively and decline over aging. They're significantly reduced with many different clinical populations who have cognitive impairment. It tracks consciousness with anesthesia and disorders of consciousness. You can see it come and go. And then it also predicts individual fluctuations of mind wandering. So we are interested in looking at many different interventions that can, basically mitigate this hyperconnectivity. It can be done with, ECT, TMS, ketamine, antidepressants. All of these have been shown to downregulate that hyperconnectivity, but it can also be done with exercise and mindfulness. So for the early detection work we're doing, we're trying to identify these biomarkers very, very early. So for instance, when people come in to as adults, they have the subgenual DLPFC that localize the DLPFC that's maximally anticorrelated subgenual, target that for curing or helping with depression, mostly driven by diverse breeders. But that same biomarker can be seen in teens. That predicts anxiety and depression, and 8 to 12-year-olds predicts 3 years later, maybe conversion of depression. And we can even see that same biomarker in young kids that predicts worsening of anxiety and depression 4 years later. So I am going to skip through the others and just say that I want to thank all of you very much for your development. I want to thank my lab and all of my clinical collaborators. John will be hearing from next, and Alfonso, who's developed the CON toolbox, and to Carl and to all of you for your phenomenal work, which has potentiated all of the research that we do. Thank you.